you, but hopefully some other time. So
Yes.
hopefully at least we'll get the science today, and then we'll get to meet at some point.
Okay.
But thank you again for the opportunity, and I'll tell you a little bit about some of the work in our lab. Essentially, what we are trying to do is take a technique that is sort of a workhorse of analytical chemistry and try and turn it into a routine instrument, an imaging instrument that can be used widely by a general audience in other fields of science. So I'll give you an example. What you see on this cover page is actually a fingerprint. And this fingerprint is not made by using black powder like you see in CSI, right? Where they go in and sprinkle something and put a tape and then pull off the tape and you can see the fingerprint. That's wonderful technology, but it destroys the print and it just destroys the evidence in the print. A really competing technology to understand what's inside the print, for example, is in the airport. So if you've flown lately in the last few years, if you go to the airport, sometimes they pull you randomly aside and they take a little cloth in a wand and they run it over all the surfaces of your suitcase. And so they gather evidence, right? But in the process, they destroy all the fingerprints that are on your suitcase. So many of the evidence gathering processes that we have are destructive and they're invasive and sometimes affect the sample itself. So that's where this technology that we're going to talk about today comes in, because this print was created using light alone. The blue color you see there is based on the concentration of oils that are in your hand. So if you touch your face, for example, you transfer a little bit of oil from your face, and the next object you touch, that oil gets transmitted or gets deposited on that object, right? So all our hands have some natural oils that we leave behind every time we touch something. Now, there are some elements in here. You can see towards the middle, you can see a bright green sort of object, and you can see small green specks everywhere else. So the bright green object or the greener object is sort of a fiber. That's a fiber maybe from the clothing that was handled that was close to your hand. And the green, the chartreuse kind of flakes are skin flakes. They're essentially protein-rich materials that were deposited along with your fingerprint. So that's all natural. The blue is the oils in your hands. The green, the chartreuse color, are the flakes of skin that come off your hands every time you touch something, right? The red pixels now are more interesting. The red ones are a trinitro compound that forms the essential element of RDX, which is an explosive that is used to demolish things, right? So ordinarily, one will not have the red compound be present in your hands. And in this particular case, if you look at the amount of compound on one pixel, that is the amount that would be left in your hands after you touch a surface 50 times after handling RDX. So the point that is illustrated here is that you can make some really sensitive measurements. You can make them non-invasively, and you can make them with a chemical basis rather than a morphological basis, or just a purely morphologic and not chemical, or a purely chemical and not morphologic basis. And at the end of the day, what we want to do is figure out if there is something bad going on, which means there was an explosive in this particular image, and we want to figure out who did it. How did it happen? So like we see the fingerprint here, we can take this fingerprint and match it up against the FBI database, and we will get the identity of the person. Similarly, can we look at disease and look at its fingerprint, match it up against a database, and get the reason why we're seeing it? Can we find those little red specks of disease and actually find whether they're going to be dangerous or not? Okay, so that's essentially what this is all about. And by the end of the talk, I'll show you how we can engineer a particular process to make this happen for different kinds of applications. So one of the applications that we're interested in that we're going to talk about today is, can you see my slides moving?
Yeah, your slide move, it's now, yes.
Now you can see it. Good, I just wanted to make sure.
Yes.
So what you can see there is the way almost every cancer in the body is diagnosed. Any cancer that is not in the blood, they take a piece of tissue out, they stain it with dyes, look under a microscope and you can actually see they use a pen to mark the image, right? So here, these two parts onto your left are ones where there's cancer. The one in the round circle there is normal tissue. And the way this process works is that you need a dye. You need a person to come in and look at it in a microscope. The molecular specificity of the dyes is not particularly high. You know, in this case, the two dyes, one goes and binds to proteins, the other binds to nucleic acid. So it's not a particularly informative dye. You can certainly, you've put two dye colors here and tested two classes of compounds. If you wanted to test more compounds, you have to put more dyes. And at some point, the color just sort of blends and you can't do much more than a few dyes, right? So you can't do more than a few colors, which means you can't really measure more than a few molecules in this case. And finally, you need to go to medical school to be able to do this, right? You need to spend 4 years in medical school, and then you need to become trained as a pathologist, and only then can you figure out if there's cancer or not. What we're trying to do is change this. And instead of this kind of view, we're trying to present this kind of view. So the middle image you see is just a compositional image. So chemists most often say, oh, I have compound A and compound B in my mixture, right? What you're trying to resolve here is what kind of cells you have in your mixture. So the middle image is just telling you that here are epithelial cells, which are cells that line the ducts in our body. And 85% of the body's cancers arise in epithelial cells, okay? So it's important to find epithelial cells. So these so-called stromal cells, or stroma there, are all the other cells combined. So they might be blood vessels, they might be neurons, they might be other things. So we're not gonna be concerned about them because in this case, this is a prostate tissue sample, and more than 97% of prostate cancers arise in epithelial cells. So we're gonna forget stromal cells for a second and go over to the third image that is to your right. And what we've done here is turned off all the stromal pixels. So the pixels that were all purple have disappeared, or that were this pinkish purple color have disappeared. And now we're left with only epithelial pixels, and I'm recoloring them. I'm leaving the normal pixels green, and the cancer pixels I'm coloring by two other colors, right? So you can see right away that I can easily pinpoint where the cancer is and where the normal is. And I can compare it to what my pathologist was saying, and I can see, in bright color, what everything is. So the way I'll show you in a few slides, the way we do this is instead of dyes, we use spectroscopic imaging. So we're not actually staining this tissue at all. We're just going to use spectroscopy. Instead of manual decisions, we're going to use computing. Instead of being not very specific, we're actually going to use spectroscopy. So we'll be very, very sensitive and specific in terms of molecular content. Our technique is non-destructive, so you can take the same tissue sample and do other things to it. And finally, you just need to be able to see essentially colors. You don't need to be an expert in the domain, and we can tell you, for example, in this image, yellow color is grade 3 cancer, so it's very easy to see where there's grade 3 cancer and where there's grade 4 cancer and where there's normal. You don't need to go to med school to interpret this image. So this, most of all, we think this technology has 3 advantages. It's label-free, which means you don't need any dyes, which means you don't need reagents, which means you don't need a cost associated with them. Second, it's molecular rich, which means you can perhaps do more things than is possible in pathology right now. And finally, it's all digital, which means you can put it into an instrument, press a button, and you don't need to do anything after that, right? So it's a potentially really convenient technology. So how does this magic all work? Well, here's the traditional way. You take a sample, you stain it, look under a microscope, then the pathologist tells you what's going on, right? What we are going to do is take the same sample, no need to stain it, but put it into a spectroscopic microscope. So this is, you can see in the middle, essentially this form factor looks like a microscope, and the thing in the foreground is a spectrometer. In this particular case, we're depicting an infrared, Fourier transform infrared spectrometer. So this is something that you use in chemistry 101, right? You go and learn about chemical analysis. So it's a really simple idea to combine these two things, but I'll show you in a second why it gets really complicated. And then at the back end is computing, right? So there's a computer lurking in the background, sort of stalking the other two instruments, if you will, because that's really needed. What we get out of this data is actually X and Y. It's the same X and Y that you see in this image, the flat red, green, and blue image on the left. But here now, at every pixel, we have a full spectrum. So those of you who remember chemistry lab, this is a standard infrared spectrum. On your right side is the shorter wavelength, about 2.5 microns in wavelength, 4,000 wave number. On your left side is the long wavelength region, about 10 microns in wavelength, and about 1,000 wave number region there. So that's interesting. I can go to any pixel and pull out a spectrum. I can also sit somewhere on the spectrum and plot the absorbance over the whole image, right? So here are a couple of examples. So on the left-hand side in the image, I'm sitting on a collagen-specific peak, so I can plot where all the collagen is, where all the protein-rich material is. And on the right side, I'm sitting on an epithelial cell-specific protein, and that's I can map out where all the epithelial cells are. So if you were an expert, again, if you went to chemistry school, you might be able to recognize what to do with these spectra, but most people cannot. So what we do then is we build computer algorithms, artificial intelligence algorithms, and we can present colors that look like this. So you take machine learning data and machine learning algorithms, and you can show where every cell type is, right? So what we've done is taken unstained tissue and put a color on it, and that color has meaning based on pathology knowledge or biomedical knowledge. Okay. So what is this good for? Well, the first thing you can do is you can be very efficient in discovery, which means you can do research much faster because now you're doing automation. So just like many factors in our life in which automation reduces cost and speeds up the process, this is what it can do. The second, perhaps more relevant to clinical operations, is that we can prevent errors. So imagine if you had an expert always with you to help you do your homework, right? You would make very few errors then because the expert could help you and guide you. So we think this system can act as an expert to help do that. Third thing we can do is, many times our knowledge is limited about how cancers behave. So could we diagnose cancer better because we have the power of artificial intelligence, right? More importantly, could we provide a level of care maybe that is available at Mayo Clinic or Johns Hopkins University where we live? So could I, in Urbana-Champaign, which is a small town, could I attract really high quality pathology assistance based on this kind of algorithm and this kind of instrumentation? And finally, I'll show you some examples. One of the biggest problems in cancer today is that when cancer is diagnosed, we don't know if it truly is a really dangerous cancer or if it's not all that dangerous, right? If it is very dangerous, we have to treat it very aggressively. If it's not going to be all that dangerous, then we're not going to treat it that aggressively. We're maybe going to treat it a little bit, watch what happens, and then treat it some more, right? So the precision in treatment, which will avoid toxicity, which will avoid injury to the body, can come from this. So the first part of it, efficiency, is fairly a lower benefit per se because it's not life and death issue. You're just doing things better, faster, cheaper. The last thing, prognostication, actually is life and death, right? You're trying to predict what is the right therapy at the right time so that we don't waste time in getting the right treatment to the patient. So I'll give you examples of several of these, but they're all published examples from the lab, so you can find many of these papers online. So we tried to, let me start from the bottom. Let me start from the prognosis problem in how we talk about today. And it's not as simple as saying, let's just image it and we'll figure out prognosis. What we decided to do a few years ago was take a little different view of cancer. So cancer traditionally is just the uncontrolled growth of cells, right? It's just cells of one type where the cancer cells and the cancer cells are growing. That's the traditional view of cancer. What is emerging now is a much more sophisticated view, right? Cancer doesn't grow by itself. It needs blood vessels, for example, to supply it oxygen because it's growing fast and nutrients because it's growing fast. A growing cancer also attracts immune cells. So different immune compositions in the cells happen. A growing cancer also elicits a response in the body. So you see different kinds of cells called fibroblasts or activated fibroblasts around a cancer, right? So cancer is not a single kind of cell growing uncontrollably. It is a single kind of cell growing uncontrollably, but supported by many other cells, right? And if you cut off the support, or if the support is wrong, then the cancer cell will not grow. Okay, so that's the hypothesis. Now, this is not new, right? People have known this for 100 years. What is new is that measuring the microenvironment was really difficult, right? You had to put in many kinds of dyes, and you had to then make measurements many, many times over. So what we are doing with spectroscopic imaging is we're measuring the whole tissue, including the microenvironment, and then we're using machine learning or artificial intelligence to figure out which part of the microenvironment it is and whether it's activated or not. Okay, so that's the first idea, the first concept. The second concept is actually more interesting. It's actually in the palm of your hands. So everyone pretty much today has a smartphone, right? And when you type on your iPhone, for example, how, it suggests, are you, right? If you type, if you ask your family member, how's mom, pretty much every day in your text messages, it learns over time. And so the next option it will, when you type how, it will suggest to you is mom, right, as the next set of suggestions. So this thing is called frequent pattern mining in language. And it's not as simple mining as which word comes after which, because there's a syntax, right? After how, you expect is, or how are, or how will, right? There's only some words that you expect after how in general language usage. So there's a constrained list of possibilities that can happen, but there's still several possibilities that can happen, right? So we took these ideas and this algorithm from natural language processing and applied it to spectroscopy. And again, this just schematically shows you how it happens, but essentially think of one spectral quantity and then the expectation of a second spectral quantity with a certain probability, and a third and a fourth and a fifth and a sixth. So over time, we start getting more and more information about a certain condition. And with a frequent spectral pattern, I can tell you if something is associated with disease or not, right? Remember, it's not causation, it's association, and then we'll figure out causation in a second aspect. So quite interestingly, we applied this to disease. We tried to distinguish dangerous cancers from non-dangerous prostate cancers. So prostate cancers, the dangerous ones are the ones that come back. So you take surgery, you take the tumor out, and then a few years later, the person has cancer again, all right? Or you treat the cancer and a few years later, they have cancer again. So we asked the question, what is the difference between a cancer that comes back and is of concern versus a cancer that will not come back, right? So if it's just a cancer that's not going to come back, then we're going to do regular surgery, tell the person to go home, good luck, everything's going to be fine. If the cancer is going to come back, then we have to treat them very aggressively and follow them very aggressively. They need to come see the doctor every month or every 3 months or whatever time, right? And so we try to figure that out. So what we did was we measured tissue and we put it into our algorithm. And we said, go forth and find the areas and why some cancers are dangerous than the other, right? So our algorithm picked up a few areas, and I'm just showing you some examples here. But let me show you the results. So on the top is the prediction from our algorithm. The right-hand side is the fourth quartile, which means the most dangerous cancers. The third one is the next dangerous, 2 is next dangerous, and 1 is the low danger, right? And you notice that the IR method, which is our method, predicts a score. The larger your score, the higher your probability, the higher your risk of having dangerous cancer. And then the next 2 rows compare our results for the exact same patients to 2 methods that are used in the clinic right now. And those 2 methods, don't do such a great job. And you can see that it's a difficult problem to do. And our method actually can provide a better aspect to it. The reason our method can provide a better understanding is because we are including information from the microenvironment, and these 2 methods are not including information from the microenvironment. So we took this step, we took this idea one step further, and I hope this paper is accepted soon. We've sent the revised version in. What we did was we took 800 colon cancer patients, and we asked, could we separate the dangerous patients, the dangerous cancers, from the non-dangerous cancers? And again, we measured the microenvironment, we measured different things in the microenvironment, and we put it into an algorithm. And it turns out, indeed, we can create 2 groups of patients. One who are at low risk, which means, if you treat them, they will be okay for the most part. 80% of them will recover just fine. And the second one is if you treat them in the traditional way, only about 50% of them will recover. So again, what we would like to do is have a perfect separation between these two, but at least we obtained some separation in these that cannot be done right now clinically. So if a patient comes in and is on the high risk branch, then we would treat them more aggressively, and we would of course pay more attention to them as their treatment progresses. And the other one, we can pretty much give the standard treatment and we will get good data. So all of this is good. Let me make one point here. All of this is good, but when we talk to our clinician friends, they said, okay, if you look in the top row of these images on your left-hand side, the so-called H&E images, those are images they're used to seeing, right? And if you look at the IR images at the bottom, they're all color-coded. So we're showing them some very colorful images and so on. And we were very happy. We said, oh, look, you can see cancer in full color now, as opposed to 2 colors in the past, right? And they weren't so excited. They said, we still need to see things because that's how we learned, right? That's our knowledge base. We learned based on this kind of image. So this new colorful image you're giving us, that's fine. But give us our old image too, so we can see, we can sort of make the correspondence one-on-one. And we said, that's great. We will not only give you the old image, but we'll try to do a little bit better. So here's what we did. And I'll cut this story short a little bit because this is now published, so you can look at the patent or the paper. And what happens in the clinic is what you see on the left-hand side, right? They take the tissue and they stain it with different dyes. We've discussed this, previously. What we did is images you can see on the right, these so-called digital stains. And what we did here was we measured spectra and used a computer algorithm to color the image now. So it gets colored just like the clinical image, okay? So if you're now hearing me, you will say, oh yeah, the colors look similar, but your digital images kind of look hazy, right? They kind of look not as sharp as you might expect. So for those of you who recognize that, hold that thought for a second. We'll come back to it in a minute. What we can do though is we can create composite images like this, right? We can make an H&E image and then we can overlay molecular images to it. And I can tell you this image on the right side that you see is completely digital. There is no stain involved in this image. It's absolutely all computation. Okay, so those of you who still remember that we were poorer in resolution, the image looked kind of hazy. The reason it happens is this, right? Here on my left is a visible image, and I've taken a piece of the visible image out, and you can clearly see nuclei, right? Nuclei are about 3, 4, 5 micron, in diameter, so you can easily see them on the stained image. And on the right is a corresponding IR image, right? And you can see that your pixel size is about the size of the nucleus, so you can't really see nuclei, really well, and the image doesn't look that sharp. And for a long time, many people in the field, myself included, said, well, okay, it doesn't look sharp, but who cares, right? We're getting all this spectral information. We will make decisions based on spectra. But actually, you are losing something, right, if the image is not as sharp. So about a decade ago, we asked a few fundamental questions, right? We said, first of all, how can we get image quality of infrared microscopy up? And second, what are really the analytical limits of IR imaging, right? How far can you push it? Could you get an image of a single molecule, for example? Could you get an image of a single cell? Could you get an image of a nucleus, for example? So these were really fundamental analytical questions. We weren't worried so much about application. We were worried about the technology and basic science and so on. So what we did, again, this is a paper that's a nice paper that's published a few years ago. If you want, take a look at it. And this is a great example of how basic science drives a lot of technology, right? So what we did in this paper was using so-called Fourier analysis, we built a complete model for the IR microscope from source to detector. There are no adjustable parameters in this. This is completely analytical, right? And at the end of the day, you get an expression, that the equation that is given at the bottom. The equation says that the electric field detected at the detector, right? That's the final thing that you want, is a function of the incident field, which means the emission of the source, as modified, as propagated through the propagation function, which tells you how light moves through the microscope, and as modified by the sample, right? So you see a sample function there, and also modified by the lenses that you're actually using, because that's what we wanted to model. We wanted to ask the question, what if we changed the lenses? What if we changed some of the parts of the optics? You know, how would our sample look like, right? And then we ran a whole bunch of simulations on this and compiled our results, okay? So on the top is a way to look at results. What it means is that if you have the same object repeated many times, which means how many line pairs do you have per millimeter, can we resolve that with a certain microscope? This is a very standard way of measuring a microscope's performance. And we showed that our theory agreed with the performance of the microscope. So that's a good sign. It's always a good sign when your theory agrees with your experiment, right? Then on the bottom, we plotted what happens if you change the numerical aperture and you change the wavelength. What is the highest resolution that you can get, right? And lo and behold, we were very surprised when we saw this. So for example, look at 4,000 wave number. 4,000 wave number is 2.5 microns, right? So compared, if you think about Rayleigh theory of optical microscopy, 2.5 micron should mean that you can only have a resolution that's about 2.5 micron, right, approximately. And when we used a high numerical aperture lens, what our plots were telling us that you can actually have a resolution of 0.6 micron, not 2.5 micron. So we said, this is really puzzling. Surely the software must be wrong. We made a mistake somewhere. So we spent many, many months trying to fix our software, right? And then we were convinced that no, it's not the software, something else is going on. So we actually built an instrument, and you can see the results. So on the left side is the commercial instrument that was operating at the Rayleigh limit. On the right side is the instrument designed with this plot that you see in front of you in mind. Here's another example of colon tissue. And you can see right away, obviously, that the resolution and the image quality is much, much nicer in this technique. And this technique we now call HD, which is high definition technology now. And pretty much every manufacturer has incorporated HD imaging into their instruments now. Okay. So this leads to some interesting scientific questions. You know, what is resolution? What magic happened here, right? How did it become, you know, how did you get such image quality? And I'll give you the short answer. The short answer is that when you think about Rayleigh criterion, what it says is that to separate 2-point objects, 2-point sources in the focal plane of the microscope, they need to be separated by a certain distance that depends upon the wavelength and the numerical aperture of the lens. Right? That's good. That's where the Rayleigh criterion comes from. What it doesn't tell you, though, is that the two sources, the 2-point sources, have to be spectrally and temporally identical. Over time and over the spectral domain, they have to be exactly the same. If they're not exactly the same, if they're slightly different, you can imagine that you can turn off one of them, right? And you can measure the other one. Then you turn off the first one and you measure the second one. And now your problem is not how do I separate 2 points? Your problem becomes how do I localize 1 point? What kind of optics do I need? Where is the signature of this one isolated point that I can do it? And this is an idea that is behind the super resolution microscopy in fluorescence, like Palm Storm microscopy, which won the Nobel Prize a few years ago. But for spectroscopy, you don't need a probe. You can turn it on and off based on which frequency you're measuring, right? And the difference in frequencies. So we said, why not design the microscope to be sensitive to that condition and not to Rayleigh condition? And that's essentially what the science boils down to, right? But you can see right away that without any, there's no computation involved, there's no simulation involved in this. This is purely collected data based on the instrument and how the instrument is set up. So you can obviously see that you know, the design of this design actually works. So what we've done since is that we have now added in to this design high spatial resolution for our machine learning. And I'll come to that in the second part of this talk for a second. But let's go back to this plot. And what this plot has really inspired us to do over 10 years is really make a march from sort of microscale to nanoscale. And that's the technique I'll tell you. So one way to improve on this plot is to simply increase the refractive index, right? So, sorry, is to increase the numerical aperture of your lens. And one way to increase the numerical aperture of your lens is, instead of having your lens be the refractive index between the lens and your sample be air, you can have some other material, like a solid immersion lens, for example. And we used a germanium solid immersion lens. And this germanium has a refractive index in the mid-infrared of 4. So you can take your numerical aperture of your lens, multiply it by the solid immersion medium refractive index, which is germanium, and now you get 4 times the numerical aperture that you were after. And what that tells you is that your pixel size can be 1/4 or smaller compared to what you were seeing before. So here's an example of below sub-micron pixel sizes using a solid immersion lens. So you can change numerical aperture, right? That's one knob you have. You can change wavelength. That's the second knob you have. So this is, I'll talk about this in a second, but we can excite with infrared and measure with visible light. So you can change the frequency from 2.5 micron to 0.5 micron, which means you can gain 5-fold in resolution as well. And finally, you can do one thing that's actually just a really nice emerging topic. You can change the detector. Instead of having a far field detector, you can have a so-called near field detector, which is really, really close to the sample. In this case, we use an AFM tip as a near field detector. And if you're interested, this is a really nice study in Nature Communications this year, where we published a whole setup and theoretical analysis that allowed us to make these measurements in the near field, which means we can get now really high resolution down to about 30 nanometer actually now from IR microscopy. So you're using wavelengths of 10 micron, and you can go all the way to 30 nanometer resolution, of course, under certain conditions that we have to carefully deal with. So here's as promised, this is some more details on the what we call infrared-optical hybrid microscopy. The idea is to excite with infrared, so you can see this this little IR beam coming through, the yellow, the one indicated in yellow, that's coming through from the side from a laser, and it illuminates the sample. The sample absorbs the light, it expands ever so slightly, and using an optical microscope equipped with an interferometer, we can actually pick up that slight change in expansion back into our system. And when we do that, we are now detecting with optical microscopy infrared light, right, essentially. And here's how it works. And here's an example of what we do with it. So again, if you can detect the infrared spectrum, you can do so-called stainless staining. And I would challenge the audience at this point now to tell me which image is real and which one is the fake one or which one is the digital one, right? It's very, very hard to tell. And unless you were involved in actually making these, you cannot tell them apart now. So we did one experiment. We took one of the leading prostate pathologists from Mayo Clinic, and we gave them 100 images. 50 of them were generated using a computer and infrared spectroscopy, and 50 of them were in the traditional laboratory sense, and they couldn't tell them apart, right? So now we have a way to do pathology with only light and a computer program, okay? So we moved on, and I just want to summarize this before I tell you a little more exciting details on how we're actually doing this. If I were to summarize this now, with IR resolution, the high definition option gives you a little bit better image. If you use optical microscopy, you can get sort of optical resolution. If you use an AFM, you can actually look at subcellular, maybe perhaps even single molecule one of these days resolution. And we actually have done some theoretical work a few years ago where we can go super resolution, which means we can go even better than 10 nanometer theoretically right now, but hopefully we'll be able to implement it and actually get there at some point. So let me switch gears quite quickly and tell you about another interesting thing. And the interesting thing is how much data is enough, right? When will the madness stop? We can keep increasing the resolution and so on, but at some point we have to say, look, increasing resolution costs me more money. I have to spend more time to collect the same data. I have to deal with more data. It costs, there's a cost associated with more data. So the question is, what kind of outcome do you want from the data? And then you adjust your resolution or your equipment to give you that outcome. So here's how we do it. We take our acquired data, for example, with FTIR. We find out which parts of the data are useful in prediction. We compare that to what is done in the clinic, for example. And then we take that smaller data set and we build instruments to actually just measure only the data that is required. And as opposed to Fourier transform infrared, which measures the whole spectrum all the time, we invented this technique that we call discrete frequency infrared, which only measures some frequencies that you need, essentially, and not the whole spectrum, right? So this is truly chemical imaging, not spectroscopic imaging in that sense. And our first attempts to do this were now in hindsight somewhat comical, but at that time we thought it was a great idea. So what we thought was we would build these very narrow reflecting diffraction gratings, essentially. And using modern fabrication techniques, we were able to do that. And if you see down here, this right at the bottom, this disc-like object, we put in many filters on this disc, much like a CD. This was a single wafer, and this wafer would rotate. So when it was rotating, it would throw off a narrow band or a single frequency of light, and we could take that light, put it through the microscope, and then we could get a chemical image, of course, of that. The problem was we took a really weak broadband source here, and then we threw away most of the light and only got a little bit of light through. So that meant that we got images, of course. Here's the data. We were able to implement this. We actually made the instrument and we implemented it. And of course, you're able to get images like you see down at the bottom. But as you can see, they were extremely noisy because you just didn't have enough signal, right? You threw away most of your light. Another thing that happened about the same time, so this was 2010 when we built this, this really nice reflecting discrete frequency system. 2011 or so, quantum cascade lasers became available. Now, this is a really fun tool because you can tune your frequency of the laser to be at any of the mid-infrared frequencies, which meant I got the super high intensity of a laser, but I got the tunable capability of an interferometer. So that's great because I can combine really high intensity into instruments. So we built the first instruments about 5-6 years ago, 7 years ago now. And I just wanted to share with you the progress in the instrumentation, right? So if you go from 2019 to 2020, you'll see that not only can we image a larger area in a much smaller time now, but the image quality has dramatically improved because systematically we've solved one problem and then the next problem and the next problem and the next problem and so on. So it turns out today you can get absolutely pristine data with microscopy that wasn't imaginable just a few years ago. We're now doing things to make this even faster. For example, here's a plot that tells you compared to FTIR, how does this microscope correspond? And just to give you a sense for those who are buffs of analytical instrumentation history, FTIR was such a fabulous technology, such a fabulous invention that by about the early 1970s, it had killed off any other way to do infrared spectroscopy, right? Whether it was gratings or prisms or anything, it was all gone because the FT process was so efficient, all right? So to my knowledge, this is the first instrument in 50 years that shows a higher performance than FTIR. And it's all because of this laser instrument that is laser quality that has come out. Anyway, for us, this is really exciting because we can get high resolution imagery. So you can see on this bottom right, we can image single cells and compare them to fluorescence, for example. And then you can see to your left, we can also image extremely large surgical sections. So this is almost like a gigapixel image now of a whole surgical resection, and we can do all of this fairly quickly. So we built instruments to do this, and I want to share this result with you. So this particular instrument that we built and reported a couple of years ago is able to, in 30 minutes, take a patient sample and tell you if it has cancer or not. So today, if you take a sample from a patient, it goes to a pathology lab, they process it, 24 hours later, someone looks at it, and in 2 days or so, you get an answer, right? So imagine a patient who's undergoing a biopsy for breast cancer. They just had a mammogram. Someone stuck a needle in them, took a piece of tissue out, and said, okay, go home, we'll tell you in 48 hours or more what happens. What they could do instead is take that tissue, since we're completely non-destructive, put it on our instrument, get an image like this in 30 minutes, and before the patient has left, tell them, there's nothing to worry about, it's all clear, or say, unfortunately, we have to get you in for treatment very quickly because we're not going to waste any time going forward, which is what the patient would want. Here's some more examples. You can now see, as the way I see it, cancer in color, right? You can actually see it in all its glory there. We can also translate the same technology to look at very large surgical samples. And what we're trying to do now is see if this technology can be implemented in a surgical theater. So when a patient is being operated upon, you can make sure that all the cancer is out and nothing is left inside and help that. So here's just an example of how this might work. So the tissue comes out, it goes under a microscope, and you can almost see the microscope, in this case, adding color, going from bottom right to top left. And while it's going, it's also labeling things, right? It's saying where there might be some concerns of interest, it's going to label them. And it makes it really easy for pathologists to appreciate where things are in this case. I just wanted to tell you again, since there are students of analytical chemistry here, what we have done at the back end of this is made a very careful trade-off. And this trade-off is central to all analytical chemistry, right? You can get very high accuracy if you have very high signal-to-noise ratio, or if you have a ton of data, if you have a lot of data. Once you start to cut down your data or your signal-to-noise ratio, you increase your speed, which means you can get the result very quickly, but you're not as confident in the result because it's much more noisy now. So each one of us, when we design an analytical solution, has to trade off an acceptable level of error compared to speed, because we have to give answers in a finite time. Okay. So the way this equation has fundamentally changed about speed versus giving an answer in finite time is the advent of deep learning and the advent of machine learning in general. So again, I won't go into it. I'll give you 4 examples. The idea behind these examples is you can have a machine learning method in which you understand what's going on, like a Bayesian method, which explicitly tells you there are 2 quantities, I'm separating them, this is the probability overlap, and you can understand this. And at the other end of the spectrum, many of you may have heard the so-called deep learning, right? You may have heard it in the context of deep fake videos that go around Facebook, etc. But this is deep learning, which means it can learn the structure and reproduce the structure using machine learning, using computing, but we can't interpret it. It's not easy to interpret why we're getting the result we're getting. So in our lab, we use a lot of these techniques, and I'll just share with you some examples. So here's an example in which we are trying to classify an invasive tumor from ductal carcinoma in situ. Ductal carcinoma in situ is a condition of the breast in which the tumor is confined and probably will not go on to metastasize and kill the patient, right? Whereas an invasive tumor, that's an aggressive tumor that's going to spread and lead to death. So using deep learning, we were able to separate these out quite easily, and you can see on the chart on the right how well they're separated, close to 100%. Another way that we're using sort of the real fake idea of deep learning is to actually generate artificial tissue models. So computational models in the tissue that can be helpful to train not only pathologists, but also computer algorithms. So what we've done is we've made one model to generate different kinds of structure from what we know about the tissue. And then we've generated another computer program to tell if that is a fake or not, right? And by racing against the discriminator model, which means is it real or is it fake, the generator model becomes better and better, right? And eventually it becomes better to the point where it can fool the discriminator model. And hence we have a fake representation that actually looks like real. And just to give you a power of this, so this is, we're trying to image a brain here. So this is the usual stuff. On the left, we have the infrared image. And I want you to pay attention to the rightmost column, which says CNN, which is convoluted neural networks. So this is a traditional machine learning approach. And you can see that, the image compared to the middle, which is the actual chemical stained image, the Nissl stain, the image isn't, loses a little bit of detail. With deep learning, we can recover the detail, right? So this is traditional machine deep learning. Traditional machine learning, and then deep learning, right? So we've now come to very powerful methods in which we can recover, we can get at just using light and a computer program what the tissue morphology looks like. Okay. So I'll skip over this, but there's some cautions that I want you to exercise. So quickly in the last minute or so that I have left, I want to emphasize a couple of things. One, as an analytical chemist and in chemistry, in your careers, you're going to see this greater integration of instrumentation and machine learning. This is a trend that's here to stay because it offers a number of advantages, not only making your instruments faster, making them more accurate, and at a lower cost. And certainly the traditional idea of measurement science, I think now has to be thought about in measurement and data science sort of combined. But I also want to challenge you to imagine new places in which this measurement science can go, right? So here are two ideas to think of that are broad. One is that there is already between measurement science and data science an exchange happening, a synergy happening that is driving analytical chemistry and analytical science broadly, and that's going to continue. The second, of course, is that even when you have this synergy, even when you have this great exchange, you need a domain partner. You need medicine, for example, otherwise you'll not be able to translate it into medical use. So what we've been doing at the University of Illinois is trying to be a good partner to translate measurement science. We were traditionally very strong in analytical chemistry and measurement science, but lately what we've done is focused our attention on cancer. And cancer, for those of you in the audience, many of you will be aware, is a really big problem, not just at the individual level, but for society. And you can hear some numbers, you can look at them while I talk, but currently there are about 18 million worldwide cancer cases expected to grow in just about 20 years to nearly 30 million people, right? So this is not only a problem, but an increasing problem in the world. It's also a really devastating problem, right? Because it's sudden, it's random mostly to people, and the prognosis is uncertain, and it has a very high frequency in our modern day society. It has major disparity concerns in socioeconomic backgrounds and how people are offered care, right? So these were the motivations, how can technology level the playing field? That led us a few years ago to establish what we call a new kind of cancer center. And the mission of this cancer center is to really initiate and translate basic engineering innovations of the sort that I've shared with you. And at the University of Illinois, bring them to bear on cancer as a problem. So we have 6 teams in our cancer center that you can see over here, ranging from drug discovery to imaging to computational biology to engineering tumors in the lab. And we hope that by putting all these together, we have a better future tomorrow, driven by science, driven by engineering innovations that allow us to take care of people much, much better. And as you go forth in your career, I would say this exchange between the sciences and the health of people and their well-being is only going to become stronger, and we all should pay attention to it for all the right reasons. So I want to stop there. I just want to thank all the people in my lab who did a lot of the work that I showed you today, some of our funding sources, and thank you so much again for having me here for this seminar. Thank you.
Thank you very much. So some questions have been already posted, and I'm going to give the students who ask the questions the honor of asking it themselves. So I'll start with Nick. Nick, you could unmute and ask your question. Where's Mick? Nick, sorry, Nick. Here's Nick. Is Nick still there? No. Okay. What about Anaya? You guys could unmute.
Hi, good evening. So you mentioned that the first instruments in that this was that you helped create the first instrument in 50 years that was able to give a higher resolution than the FDIR. And I was wondering, weighing how much it would cost and the data you would get from it, how much value would be derived from creating an instrument or adjusting the instrument so it would give even higher resolution?
Yeah, that's a good question, Anaya. So the you bring up an important point, right? That we can make advances, and somebody has to eventually pay for it because it's useful for them, right? It brings value to them. And there are two aspects. Sometimes you do science for the sake of advancing scientific knowledge, right? And with no real applications in mind, and then eventually it finds an application. In this case, there are some really definite points, right? So for example, a resolution of 0.5 micron is typically what is common in analytical, in measurement in clinical samples. So that was a great milestone for us, right? And a resolution of, say, 10 nanometer is really useful in looking at single protein, single molecule samples. So that getting to that level is also another milestone, right? So there's a synergy between applications and instrument design that really makes it successful at some point. So, there's a push-pull, right? The technology is available, people find use for it, or they ask a certain performance and we develop the technology. It works both ways.
Okay, let's hear from Daniel. Daniel Chi has a question.
Hello. So I came in a little bit late because I was in lab. My question, I think from what I understand, it uses computer machine learning, right? So I'm assuming it like sees a case and then it kind of makes a prediction. So my question is, because I think you said at a portion that there was it had like 80% chance of this case being less severe and 50% chance of it being very severe. I think it said something like that. If it sees enough cases, do you predict that it would actually become very, very accurate? Or is there like a chance where like it might get distracted like a red herring and lead off to a wrong diagnosis kind of thing?
Yeah, I think that's a great question, Daniel. So there are safeguards built to include data into ones that can be used to improve the prediction. And once you build that in and you qualify the data you use to build your models, either by human supervision or other quality checks, then over time the algorithm gets better. No question about it, right? The more samples you give it, the better it gets. And you can train it under certain circumstances to recognize, anomalous samples as well. So the rare samples or the unusual samples, you can certainly recognize those as well. But over time, it gets better. Yep.
Okay. So let's go with Lizzie Borton. Lizzie Borton, you have a question?
Yeah. I was just wondering, since your method generally gets more detail and more information about the samples, do you think that could be used for cancer prevention research as well as what you're doing?
Yeah, that's a great question, Lizzie. So one of the things that we've lately focused on is looking at lipids, right? Looking at how lipid function, lipid transport, lipid signaling modulates the conversation between the tumor and its microenvironment. And we're not alone. There's a whole bunch of people looking at this. Where we have an advantage is that we can watch it in action. So we can actually, without labeling, we can actually see lipids because lipids have a unique chemical signature. We can watch whether the cell is turning cancerous or not. So I did show some data here, but one of the, because this was more of an analytical chemistry seminar, one of the things we're doing in my lab is we've developed 3D printing approaches that we're trying to make models of tumors in the lab so that we can watch this and then eventually translate that argument to how we might prevent things in actual patients. So stay tuned. That's a great idea.
John Wan, you have a good question here too.
You mentioned that deep learning was used to predict and produce higher resolution images. How does this transition to other analytical devices like nuclear magnetic resonance?
Oh yeah, John Wan. This deep learning is everywhere now. used in every single instrumentation. So for MRI, for example, the way deep learning is being used quite nicely now is that it can speed up the data acquisition process, because you don't need to collect all the data, right? You collect a certain subset of the data and use deep learning to fill in the blanks. Sorry, did someone have a question?
Okay, Joyce, you have a very interesting question. Joyce?
Oh, I was wondering if like this technology, since it would make it easier, like do pathologists really need to go to med school and learn stuff like?
Okay, this is really interesting, Desmond. So thank you. I had semi-guessed what you will say. So let me rephrase your question, Joyce, in another way. Does this make pathologists obsolete, which means machines will do the work and so on? The short answer is no. The way I look at it is that, you know, compared to Microsoft Word that you use today to edit documents, compared to typewriters, right, where people would type a word and it would print it on the paper, and you couldn't correct it. You couldn't go back. You couldn't refer to things. You couldn't look up the synonym for a word, you know, on your typewriter, right, which you can in Microsoft Word. So I liken this technology to be working with the pathologists and not to replace them. The reason is that, you know, you need to be absolutely fail-safe in this case. You need to really relate for many reasons. You need to relate to the knowledge base of pathologists. You need to have responsibility taken by someone to say, oh yeah, I assure this, right? So I do think that there will be a human element to disease diagnosis, simply because we should not, I don't believe we should turn to machines all over in that sense. So there will be a human element for a variety of reasons, including oversight, including simple politics, right? There's a pathology department and a pathology association. You're not going to let this happen, right? Second thing. And finally, you know, here's my personal belief. I think medicine needs to become more humane, right? Medicine needs to become more empathetic. And the reason it is not is because we're asking medical professionals to do a lot more that isn't related to what they're supposed to do. They have to fill out many more forms now. They have to do a lot more regulatory things that they weren't practicing, say, 50 years ago. So many of my medical colleagues who say, you know, I got into medicine to serve people, but what I'm doing is filling out forms, right? So we hope that technologies like this can reduce the burden on people so that they're freed up to really think about their samples and to really be empathetic with patients when they do encounter patients, right? So for a pathologist, for example, instead of spending their time trying to find the tumor on the slide, this technology will find it, put it up on the board, and the pathologist can really think through the case, right? So instead of spending 30 minutes searching for the tumor, they can spend 25 minutes thinking about what the tumor means and drawing on their knowledge and looking at that particular case of that particular human and giving better decisions. So I think that's how I see this technology being used.
Okay. Isabella, you have a good question there too?
Okay, so my question was, what properties do the machines use to detect the different types of tissues? So basically, how do they learn, and like what properties, chemical or physical? And how high is the possibility that the machines will misdiagnose or show the wrong type of tissue?
Yeah, that's a great question. So there are two things, Isabella. One is the spectral content, right? So think of the spectrum like a barcode. So when you go to the grocery store, has anyone here experienced the wrong product being scanned? I'm not talking about the price. The pricing has to do with the database, but the recognition of the product, right? If you scan a bag of rice, it will always say rice, right? So as long as you have a clear barcode, right, you will not make a mistake. So think of the spectrum as a barcode, but with deep learning, we're adding spatial organization as a barcode as well. So the level of safety with some of the modern algorithms has gone up because the size of the barcode has become really large, right? Now, the problem is, if we have not seen that barcode before, right? So if you go to a grocery store where that product is not in the database and you try to scan the barcode, it's going to give you an error, right? So the limitations of this kind of technology are primarily arising from we may not have seen cases like that enough. So over time, this technology will become better. And at some point, it becomes better than human capability. So if you look at the malpractice literature, humans are correct about 99% of the time in pathology, right? So what we need to do is we need to make an instrument that is at least 99.9% accurate, and then not worry too much about the error, but over time keep reducing those infrequent errors also at some point. Right?
Duyan, you have another good question. Duyan?
So how soon do you expect infrared imaging to become the main form of imaging?
I hope very soon, because that would be very satisfying. But you know, there's a couple of things, Dion. Maybe I'll explain the pathway in which these things work, right? So what needs to happen first is that we are an academic, right? I'm a professor. I do not make instruments. I'm not a manufacturing facility. So what we need to do is translate this technology to industry first. And you can see the logo of Agilent here. Agilent is a very nice company out in California, based in Santa Clara, just down the road from Apple. And Agilent is a $4.5 billion instrument manufacturer. So what Agilent has done is taken some of the designs from our lab and made a commercial product out of it. Now we're working with Agilent to turn that commercial product into a pathology-specific product, right? So first of all, that product has to be available. Second, we have to get a group of people to use that product on a trial basis and do a multi-site trial. Once that trial is done and the results are satisfactory, then we have to go to the FDA and say, look, the results, we did this trial, the results look great. Please allow us to make a commercial product that can go and be purchased by a hospital. Then we have to convince a company like Agilent to make a commercial product and offer the sales and service and all of the, everything that goes along with it. And then hope some pathology departments buy it. And when the pathology departments buy it, then of course you have it in practice, right? So there are about 5 steps that we have started the journey on, but I think you're easily looking at about a decade of development between now and when it's actually used.
I think Aaron has a related question. Aaron?
Yeah, so my question is I had 2 separate ones. So the first one is who developed the ideas for this infrared optical hybrid microscope? And you mentioned that you are working with Agilent, but has anybody formed any new companies with the technology? Because I know that there's a lot of potential to form a new company instead of maybe giving it to a large company that already exists. Although, there may be some like monetary concerns where you do have to do the trials and such, but are there any new companies who made those ideas? We'll just start with those because those are maybe some.
Yeah, that's a good question, Aaron. So, you often struggle with this, right? There's two things. Large companies are slow. They take their time. Their decisions are dictated. It might change overnight, right? On the other side is a so-called startup model, right? Where you take technology from the lab and you try to build an instrument and set up. In a startup model, it's much more difficult to offer sales and service, right? This is not an app where you can create it in your basement and upload it and people can use it. There's servicing required. There's technology training for the users required. So having a startup company is much more difficult, and we are thinking about it. So the IROH, the hybrid microscopy is not yet translated. That's an invention from our lab, and that's not available right now commercially, of course. So we're thinking on ways of getting that out and that being available commercially. It might involve starting a new company.
Okay. You want to do the second part of your question, or?
Yeah, so you might have also, I mean, you touched on this a little bit too, that it doesn't replace pathologists' jobs, but rather works in concert with them. So would you foresee a maybe a analysis in the 30 minutes after or so, or getting an image, and then actually since the instrument is non-destructive, then you could actually send the whole tissue as well to a pathology lab, so you could have like a doubled up analysis?
Yeah, that's a good question. So what we think, at least initially, that would have to happen, right? Because the tissue then maintains the standard of care. We just come in, we image it with light, and we're done. And then the tissue goes off to the traditional sampling process. So actually, that is one of the really important advantages of this technology, is that introducing it, even initially, will involve no change in the standard of care, right? It can only improve from where we are.
Okay. All right. Well, I think we've asked a lot of really good questions here, and we want to wrap things up now and thank our guest speaker one more time for a wonderful seminar and bringing us here to some of the edge of where we are in analytical chemistry and the future of analytical chemistry. So thank you very much.
Thank you so much, Desmond, for having me.
Okay.
Thank you. Take care, everyone.
Take care.
Thank you.